Hi Bio 111 students, and welcome to week 3 and 4 of the class. Today we're going to be talking about a familiar molecule, water. We covered it a little bit in the last lecture, and we're going to cover it a bit more extensively now. So water is a unique molecule, and it is a unique molecule that is vital for life. And one of the things you notice right away is that we have H2O with covalent bonds between the oxygen and the two hydrogens. We talked about hydrogen bonds. These are not hydrogen bonds. Hydrogen bonds happen between different water molecules when they become near to each other. And oxygen and hydrogens that come near a water molecule also could be hydrogen bonded. So let's talk about a little bit more about water. So each molecule of water is polar. It has a slightly negative oxygen and a slightly positive charge at each hydrogen. And what that tells us is that hydrogen bonds will form if another water molecule comes close. And they'll form like this. And we denote the hydrogen bond as a dotted line because it's actually a weaker bond than either covalent or ionic. And if you can imagine, you could cover this entire board with water molecules that are hydrogen bonded to each other. And that's pretty much what it would look like if you were to take a drop of water and to examine all of the various hydrogen water molecules within that drop of water. A thing to note about hydrogen bonds, is that they absorb a lot of energy before they break. Now what that means for us is that it takes a lot of energy to heat water molecules. And when you break apart these hydrogen bonds, you have evaporation. So that means that it takes a lot of energy, a huge amount of energy, to get vaporization to occur, which is at about 100 degrees Celsius when we see boiling, and that's a high boiling point for water. Many of the other solvent solutions that we encounter have much lower boiling points than this. You think about alcohol, you think about acetone, or any of the other liquids that you commonly encounter in your daily life, they have much lower boiling points than water. And the reason for this is hydrogen bonds. Another thing that the hydrogen bonds give to water is a higher specific heat capacity. Specific heat capacity is the amount of energy it takes to raise 1 gram of water, or any other substance, whatever it is, but it's 1 gram of water here, by 1 degree Celsius. So the amount of energy put in to increase 1 gram of a substance by 1 degree Celsius, and it's very high for water, and the reason for this is because of these hydrogen bonds also. Another thing that we see with water that's kind of unique, well, very unique actually among liquids, is it has a crystalline structure when it becomes solid. We don't see crystalline structures with a lot of other substances, and so for instance any of the metals, you can think of iron, you can think of gold, any one of them. When you take a solid and you drop it into a liquid, the solid drops to the bottom right away of the container. You don't see that with ice. What happens is the crystalline structure causes the molecules to stay slightly further away from each other, and therefore ice is less dense and stays at the top of the water. Because of this, pond life is possible. If you had a solid layer of ice at the bottom of a pond, that would make it impossible for oxygen to go into any organisms living at the bottom of the pond during the winter. And but thanks to floating ice, we do see life in ponds and lakes. The specific heat capacity of water also absorbs a lot of energy, and it acts as a force for homeostasis. If you think about this, land, for instance, cools and heats at 7 times the rate of water. So that means that being near a large body of water will help to dampen the rate of change of temperature up and down, and therefore you will have, slightly cooler summers, slightly warmer winters. You won't have the extreme range that you'll see further inland. And you see that with oceans and things like that. So in terms of inside the bodies of animals like ourselves, we will see a moderation of cooling and moderation of heating due to water's specific heat capacity being quite high. So, okay, now we want to talk another thing that we get with hydrogen bonds is cohesion and adhesion. A way to remember the word you think about between two individuals that are the same, I guess co-educational, co-operation. So cohesive means the ability to stick together of water molecules. So that's their overall stickiness together. And we see this when we overfill a glass, and right before it overflows, you'll actually see the water, because it's so sticky, do something like this right before it overflows. Adhesion is water molecules sticking to surfaces. I used the example in the last lecture of a glass with the water sticking to the edges. It's why when we see the meniscus in water, we see this. In a water glass, you'll have this. That stickiness at the sides is a result of adhesion. Again, it's caused by hydrogen bonds. Another example of cohesion and surface tension caused by cohesion would be the idea that we can float something that is heavier, more dense than water on top. For instance, if you're very, very careful, you can drop a needle onto the surface of water and it will float. So that's all we have about water for now. And one good thing about videos is if you did get that, you can just rewind and look at it again. So we're going to talk about pH. Some of you may remember pH has to do with acids and bases. I always use this as an idea that helps me remember which is the lower one. A, N, B. So A stands for acid, and it is between 0 and 7 on the pH scale. And B is for bases, and that's between 7 and 14 on the pH scale. And then N is for neutral. Neutral is not at 0 as you might expect, it's at 7. So that's an important number to remember. All acids are from 0 to 7 on the pH scale. All bases are from 7 to 14 on the pH scale. And neutral is exactly 7. This is an inverse logarithmic scale, and this sometimes causes some headaches for people. So 7 is actually 1 times 10 to the minus 7 moles of hydrogen in a liter of water. So it's moles of free hydrogen ions. That, when you take the inverse log of that number, you get 7. So you can imagine this is a small number, and 1 by 10 to the minus 14 is an even smaller number. So there are less hydrogen ions, less free hydrogen ions on this end and more on this end, because 1 by 10 to the minus 1 moles, for instance here at 1, is a larger number than 1 by 10 to the minus 7 moles. So that's the reason for the confusion, that people go like, well, hold on a second, the small numbers have more free hydrogen ions than the large numbers, and they can't figure out why that is. Well, it's because it's an inverse logarithmic scale. So, along with this, you'd say stronger acids will have lower pHs. And lower pHs mean that they are better hydrogen donors, hydrogen ion donors, over this side. And on the other side, we have our bases, and they are they donate OH, which is hydroxide ions, and they are a negative ion. And you'll see the H and OH should look familiar. Well, that means that these are better hydroxide OH minus acceptors, and these are better hydrogen ion acceptors. Another thing to realize about the logarithmic scale of pH is that, for instance, 6, because we're looking at 10, 1 by 10 to the minus 6 instead of 10 to the minus 7, 6 is 10 times more acidic than 7, and so on. And so that's how it works. In humans, we have a slightly basic blood. I'll just I want to get into the color, but this will work. So, bloodstream of a human, 7.35 to 7.45 is our normal blood pH. So it's slightly basic and slightly over that side. If it starts to become acidic, there are things we can do. Our body does automatically to help bring back our pH back up to 7.35 to 7.45, and there are things that our body does. Some of those things are called buffers. So buffers are found in living things, and they aid in maintaining the pH at a near to neutral value. They absorb excess ions, and those ions are H plus hydrogen ions and OH minus hydroxide ions, and there are three parts in humans. For instance, we're going to use those human beings as an example organism. So carbonic acid is H2CO3. Is found within our bodies, and we also have another ion called bicarbonate. And if you remember from the experiment about the unknowns, baking soda, that is bicarbonate of soda. It's sodium bicarbonate, and it works pretty much as a buffer, because the sodium bicarbonate dissociates, and you end up with this bicarbonate ion. And we'll talk about that in a little bit. Bicarbonate ion is HCO3. And the third ion, or the third thing that works in maintaining pH, is CO2. So these are the three buffers we see in people. So the end result, when you have too much of one or the other, is you end up with an addition of these two. This is negative and this one is positive, and what you end up with is water and CO2. And when we increase our breathing rate, when we're running, the reason for that is because our body recognizes that there is a need to blow off more CO2, because more CO2 is being produced, because we're using this to absorb the acid that's being produced within our body. So we exhale any excess CO2 to bring ourselves back into the balance, 7.35 to 7.45. Bicarbonate is the buffer that is found in a commonly used item called Tums. I don't know if you've ever used it. It's for heartburn, and what it does is it has a higher pH, and as a result it helps to neutralize some excess stomach acid, and so it's good for stomach acid. So, let's talk a little bit about carbon. And if you're into science fiction or anything like that, you've heard the words carbon-based life form. Well, that's basically all life on this planet, from the smallest bacteria to the biggest tree, all the plants, everything in between, all the fungi, and all of us, all of the animals. We are all carbon-based lifestyles. Um, life, we are all carbon-based life forms, I should say. So, all organic molecules that are necessary for life contain a backbone made of a string of carbon. Here's an atom of carbon. Remember from last week. Two on the inside, because that's always the way we do it. And then we are left with, it's carbon 612. So we are left with 4 more electrons to put on the outside. And that gives us 4 places where we can form covalent bonds. And that is the reason why carbon, one of the reasons why carbon is such a good atom for life, is because it can form 4 covalent bonds. An example would be something we use in everyday life, probably heat the house right now, or your apartment, or wherever you have it. Methane is also known as natural gas. And there we see 4 covalent bonds with hydrogen. Don't confuse those with hydrogen bonds. Hydrogen bonds, we're generally talking about water. So, now let's talk about some functional groups. You'll see the importance of functional groups later as we get further in. But for now, I just want you to try and remember some of these names, and you'll see that they kind of make sense. So here's one. Simply OH. Now, the name for this functional group is hydroxyl, and it's because it has a hydrogen and an oxygen. Next one we have because it has a carbon and an oxygen and a hydrogen, we call it a carboxyl. And that COOH ends up really actually looking like this. Next one, it's the only one that has an N in it, and that helps you to remember its name. Its name is amino. And as you can imagine, we will be talking about amino acids later, so you're going to see this word a lot. Or phosphate, basically this phosphate looks like this. P with 4 oxygens. And last one, we talked about methane earlier. Well, this is methyl. They're usually attached to something else, and that's the reason why we have these lines coming off here. They're attached to usually a chain of carbons. But this group, this little part here, is called a methyl. That little part there is called a phosphate. This little part here is an amino. And this little part here, carboxyl and hydroxyl. So those are 4 functional groups that you have to know, and they will come up in exam 1. I can tell you that right now. So, the word organic, you've probably heard of organic chemistry, organic molecules, all of that sort of stuff. The word organic refers to living things, and it also has what all living things have in common, which is carbon. So, organic means the chemistry of carbon or the biology of carbon. So, I'm going to introduce a concept now, just so you get the idea. There are a single part of something is called a monomer. So, if you imagine a train carriage would be a monomer by itself, and a train with several other carriages we call a polymer. And you're going to see these terms mono and poly used a bit in the future in this class, because monomer is one of something and poly is many of something. So, in biological molecules, we have this situation going on. Each carbon can make 4 bonds, and it can make them with either another carbon or with a hydrogen or an oxygen. But this with the 3 carbons is called a polymer. If it was just this center one by itself or one of the ones on the sides, it would be called a monomer. So methane that we showed you earlier is a monomer of this situation, which is a polymer. So there are 4 groups of biological macromolecules that we want to talk about. This is going to be the experiment this week, and we're going to be building them. We'll get more of a handle from actually having them in your hands and building these molecules with the kits provided. So we have carbohydrates, lipids, proteins, and nucleic acids. So, they're the four biological macromolecules. So all carbohydrates, just to give you an idea, they all have the same general formula, and that is CH2O. So you have a carbon attached to a water, and then however many times that happens. So, for instance, glucose. Glucose, the N would be 6, so we would have C6H12O6. So the N multiplies, everything inside. And we have different. We have fructose, C5H10O5, and so you get the general idea. They have the 1-to-1 ratio, and each subunit, something like this, is, again, using the word mono, is called a monosaccharide. Saccharide generally denotes sugars, and so let's talk a little bit about the monosaccharides. So let's put one up here. Figure 3.2 in your book, it shows how monomers become polymers, and it uses monosaccharides becoming disaccharides. So we're going to take a look at this. So we have a monosaccharide here. It's each one of these is a carbon, because the carbons don't actually stay in long chains, it's just strung out. They generally want to come together and form a tight molecule like this. So you end up with 6 carbons here. This is a 6-carbon chain. And let's say you have this is glucose. And it happens to have the water molecule stuck here at the end. So it has a hydrogen coming off here, and an OH or HO, whichever way you want to put it, sticking out like this, and it comes in contact with, let's say, a fructose. So fructose. Remember we talked about C5H10O O5. Well, fructose looks like this. So you can see there's 5 carbons, 5 different points. Carbons are at each little point here. And it also has a hydrogen and an oxygen sticking out, and it's much the same. Actually, it's for simplicity's sake, say it goes like this. And what happens is dehydration synthesis occurs. And what that basically means is that water, think about dehydration means water going away. So one water molecule leaves, and when it leaves, you're left with oxygen by itself on the bottom. This one here, and that will become the bond that joins these two. And now you have simple table sugar, sucrose. And that is a disaccharide. So, dehydration synthesis joins things. With bonds. If I was to add water to sucrose, I can split this back apart again because I'm putting this water molecule back in, and that is called hydrolysis. And lysis as a term means split apart, so you can imagine that's what it does. It splits apart disaccharides and makes them monosaccharides again. When we're talking about sugars, an important suffix to note is this one here. You'll notice all of them have glucose, fructose, sucrose. So wherever you see this, it means sugar. And you can have a lot of these together, a lot of these monomers together, and different ones, different combinations and different amounts make different sugars. The most common sugars are glucose, we talked about, ribose, and glyceraldehyde. Figure 3.4 in the book or in the readings. You can check them out yourself. So, examples of disaccharides. Sucrose is not the only one. You also have maltose and lactose. So, I would remember that for the test. Lactose is a disaccharide. Polysaccharides can, like I said, they can be any length. You can have very, very long chains of monosaccharides all bound together by dehydration synthesis to give you this bond here. And examples of the really long chains are starch, glycogen, cellulose, or chitin. Starch is the way that plants store energy. Think of potatoes or any of the other starchy vegetables. That's their way of storing energy for their seeds. And glycogen, glycogen, is the way that human beings store energy. And glycogen is stored in the liver of human beings. So, for instance, and we'll talk a little bit about carbohydrates as food, and we break down carbohydrates like starch right away, starting in the mouth with salivary amylase. So it starts from the second that food goes in your mouth. These polymers, these long chains of saccharides, these long polysaccharides start to be broken down in the mouth with a protein, specialized protein called an enzyme, and the one that does the starch breakdown part in the mouth is called salivary amylase. and you'll see this A's on the back of enzymes. All the enzymes, you'll see A's on the back. Cellulose is another one of these long things. We really, it's actually such a tough to break down thing. It's found in grass and found in other plants that other animals with multiple stomachs and specialized digestive systems eat. We can't break it down, so it serves us mainly as a form of roughage, a form of dietary fiber. So, lipids, they are mostly non-polar, hydrophobic hydrocarbons. In the cell, lipids function to store energy, provide thermal insulation, and to provide oils for feathers and mammalian hair. Classic example is the otter. The reason why the otter is so waterproof is because of those hydrophobic oils that coat every hair in the otter's coat. We see the same thing with bird feathers, especially the aquatic birds like ducks. You'll see the water bead up on the surface of their feathers because they have specialized oils that coat them. A thing that lipids also do energy storage, thermal insulation, oils, hormone building blocks, and also found in every cell membrane. So that's all we're going to do for now. I'll conclude the rest of this lecture the next day. Thanks.